Welcome to the fascinating world of gene expression. Imagine that your DNA is a priceless, ancient master recipe book locked safely inside a grand library, which represents the cell nucleus. You desperately need to bake a cake, but the strict librarian absolutely forbids you from taking the master book out into the messy kitchen. Today, we will explore how cells solve this problem through a vital process called transcription. Building on our library analogy, let us understand why transcription is so essential. Without transcription, the genetic information stored in your DNA would be completely trapped and useless. Your cells could never build the proteins required to keep you alive. Because the master DNA cannot leave the safety of the nucleus, the cell's only logical solution is to take a piece of scratch paper and carefully copy down just the one specific recipe it needs. In our cells, this temporary, movable scratch paper is RNA, and the highly coordinated biological process of creating it is called transcription. Now that we understand what transcription is conceptually, let us look at the machinery that makes it happen. This incredible task is performed by a complex molecular machine, an enzyme known as RNA polymerase. This enzyme is highly specific in how it operates. It reads the DNA template strand strictly in the 3' to 5' direction, but it builds the new RNA molecule in the opposite, 5' to 3' direction. The resulting molecule, often messenger RNA, carries this genetic code out to the ribosomes, where proteins are actually built. This directional flow of information forms the central dogma of molecular biology. This directional flow of information we just discussed is not just academic. It has profound real-world applications. Why does this specific machinery matter to doctors and patients? Because it is a major target for life-saving medical treatments. For example, certain powerful antibiotics, like rifampin, work specifically by jamming and breaking the bacterial RNA polymerase machine. By stopping bacterial transcription, the bacteria cannot make the proteins they need to survive, and they quickly die. This cures your infection while leaving your own eukaryotic cells perfectly safe because our human RNA polymerase is structurally different from the bacterial version. Knowing how the machine works qualitatively leads us to an important quantitative question. How fast does it operate? Even though biology often feels like a purely descriptive science, the processes inside our cells happen at measurable physical speeds. We can calculate how long it takes for an RNA polymerase enzyme to transcribe a specific gene using this simple rate equation: T equals L divided by R. Here, T is the total time in seconds. L stands for the length of the gene in base pairs, which are the individual letters of DNA. And R represents the rate or speed of the enzyme. If the gene length L increases, the time required naturally increases because there is more material to copy. Conversely, if the enzyme works faster and R increases, the total time T will decrease. Before we move on to the specific mechanisms, let us see this formula in action with a concrete example. Imagine a harmless bacterium in your gut that suddenly needs to digest lactose sugar. It must transcribe the LAC-Z gene, which has a known length of 3,000 base pairs. The bacterial RNA polymerase operates at a steady rate of 40 base pairs per second. We want to find the total time required. We take our formula, T equals L over R, and substitute our known values. We divide the 3,000 DNA letters by the rate of 40 letters per second. The calculation gives us exactly 75 seconds. This means it takes just over 1 minute to produce a complete mRNA copy. This incredibly fast response time is exactly why bacteria can adapt to new food sources or dangers almost instantly. Now that we have calculated the impressive speed of this process, let us look at how the bacterial machine actually operates to achieve it. Think of the prokaryotic RNA polymerase as a highly versatile, all-in-one construction vehicle. Because bacteria lack a nucleus, their DNA floats freely in the cytoplasm, making it very easy for this vehicle to access the genes. The process kicks off with a phase called initiation. During this phase, the core RNA polymerase binds to a special helper protein called a sigma factor. You can think of this sigma factor as a GPS system. It guides the enzyme directly to the exact starting line of the gene, a specific DNA sequence known as the promoter. Once the enzyme has successfully found the starting line, it moves into the next phases. After attaching to the promoter, the enzyme unwinds the DNA and begins elongation. It moves along the template strand, reading the bases and building the new RNA chain at that rapid speed we calculated earlier. Finally, the process must end, which brings us to termination. The enzyme eventually hits a specific stop sequence in the DNA. This sequence causes the newly formed RNA to fold back on itself into a rigid hairpin shape. This sudden physical shape change acts like a brake, yanking the RNA out of the enzyme and successfully ending the transcription process. To visualize the elongation phase we just discussed, let us look at this schematic of a transcription bubble. The large gray oval represents the RNA polymerase enzyme itself, which has unwound a section of the double-stranded DNA. Notice the two DNA strands. The top is the coding strand, and the bottom is the template strand, running from 3' to 5'. Inside the bubble, you can see the newly synthesized mRNA strand in blue. It is base pairing directly with the template strand and growing in the 5' to 3' direction. The arrow at the top shows the overall direction of transcription as the enzyme moves steadily to the right along the DNA. While the bacterial system we just visualized is fast and streamlined, eukaryotic transcription, which happens in your own cells, is vastly more complicated. If the bacterial enzyme is a simple, all-in-one construction vehicle, the eukaryotic system is a massive, highly regulated, multi-billion-dollar factory. Because our cells are part of a complex, multicellular organism, they simply cannot afford to make mistakes or transcribe genes at the wrong time. Therefore, eukaryotes do not just have one RNA polymerase. They have 3 distinct types, with RNA polymerase II being the one responsible for making messenger RNA. Instead of a simple sigma factor, our enzymes require dozens of helper proteins called general transcription factors to assemble at the promoter region, often recognizing a specific DNA sequence called the Tata box. Beyond just assembling the machinery, eukaryotic cells face additional physical hurdles. Because our DNA is incredibly long, it is tightly wrapped around histone proteins, much like thread on a spool. The cell must first unpack and loosen this DNA before the enzyme can even reach it. Furthermore, once the RNA is finally transcribed, it is still not ready to be used immediately by the cell. It must undergo extensive processing to become mature messenger RNA. This includes adding a protective cap to one end and a poly-A tail to the other to prevent degradation. Finally, the cell must carefully edit the molecule by splicing out useless non-coding regions called introns, leaving only the vital instructions behind. To truly appreciate the complexity of this eukaryotic processing, let us calculate the time required for a massive human gene. We will look at the human dystrophin gene, which is crucial for muscle function. This gene is an absolute monster, with a staggering length of 2,400,000 base pairs. Human RNA polymerase II is heavily regulated and moves slower than the bacterial version, at roughly 20 base pairs per second. We use our same formula, dividing the massive length by the slower rate. This gives us 120,000 seconds. To grasp this scale, we divide by 3,600 to convert it into hours. The final answer is over 33 hours of continuous, non-stop work for the enzyme to transcribe this one single gene. This perfectly illustrates why eukaryotic genes contain so many introns that must be spliced out later. We have covered a tremendous amount of ground today, so let us review the key takeaways. First, transcription is the essential first step in expressing the genetic code, converting locked DNA instructions into a readable, movable RNA format. Second, we saw a stark contrast between organisms. Prokaryotes utilize a fast, streamlined system to quickly adapt to their environment, while eukaryotes rely on a highly complex, heavily regulated system requiring extensive RNA processing. Finally, we learned how to mathematically calculate the time required for this process, proving that the physical length of a gene and the speed of the polymerase dictate the biological response time. Understanding these microscopic factories gives us a profound appreciation for the mechanisms keeping us alive every single second. Great job following along.